それでは本日、ウレ・メランダー先生の来日セミナーに関しまして、まずは我々、ムーンショット東京大学で予算を取っておりますが、こちらのプロジェクトリーダー、南岳正臣先生に、まずはムーンショットのご紹介をさせていただきます。どうぞよろしくお願いいたします。
はい、岸先生、ありがとうございます。我々はムーンショット目標7、2040年までに主要な疾患を予防克服し、100歳まで健康不安なく人生を楽しむためのサステイナブルな医療介護システムを実現するという計画に応募させていただき、臨床生命医工学連携研究機構において、この提案、具体的には病院を家庭に、家庭で炎症コントロールということで、池浦先生、鄭先生を中心にご指導いただきながら計画書を作成し、これが採択されております。本日ご講演いただきますスウェーデンのオレ・メランダー先生は、マルモコホートスタディをリードをしておられる先生で、このムーンショットプロジェクトにおいても非常に重要な共同研究者としてご参画いただいている先生になります。それではご紹介をトーマス・スウェンソン先生からお願いいたします。
ありがとうございます。It is with great pleasure that I introduce Olle Melander, who is a professor and chair of internal medicine at Lund University, and also senior consultant at the Department of Internal Medicine at Skåne University Hospital in Malmö, Sweden. Professor Melander has received numerous grants and awards for his research. To name a few, the European Society of Hypertension's Peter Sleight Award in 2010, Lund University's Fanstrom Award in 2013. In May 2011, Professor Melander was awarded the Swedish Heart and Lung Foundation's large grant, a grant only awarded to one Swedish researcher yearly with internationally leading cardiovascular research. Professor Melander has also been awarded European Research Council's large grant, and in addition to this, the Göran Gustafsson's Prize and Grant, as well as the Wallenberg Clinical Scholar Grant, both awarded by the Royal Swedish Academy of Sciences. Professor Melander has numerous peer-reviewed research publications, exceeding over 700 in count. He has an H-index of 108 and close to 70,000 academic citations. Thank you very much for joining us, Professor Melander. The floor is yours.
Thank you very much to Professor Nangaku and to Dr. Svensson and Kishi Svensson. Thank you for the invitation to talk to you on this subject today, which is part of an ongoing collaboration between Lund University and Tokyo University, University of Tokyo. We can't. So I come quite a bit from Japan, in Malmö, a town in the southern tip of Sweden, as you can see on the dot here. I had to paint the arrow since there's a war going on in Europe, so you have to fly around the Russian Federation. But we are located in just opposite Copenhagen in Denmark, and this is where the studies that I will focus today's presentation on come from. This is a slide that shows a little bit how we think about prevention in the future. This is part of a European Research Council grant that is running currently. So what we do is, just like you can have personalized treatment of disease, we would like to, because we know that individuals are also very biologically diverse before they get the disease. So we're trying to identify specific molecular risk factors that we know are present in part of the population, but not in the entire population. And then from those people who have these disturbances to go in and try to treat specifically the molecular disturbance that these individuals have. And to do that, so this is kind of the general goal of the research. It is to identify novel genetic, hormonal, and metabolic risk factors early, of course, and to intervene with specific means to those molecular disturbances in order to prevent cardiometabolic disease. And we are working with three parts, you can say, three infrastructures. One is a clinical research unit with about 20 full-time employed nurses and biomedical analysts who do the clinical exams and imaging exams of people who come into the population studies. They do intervention studies with different lifestyle trials or pharmacological trials, and also deep phenotyping. We have a laboratory in the same university area specialized at measuring sequencing and genotyping, genetics, in other words, measuring various cardiometabolic hormones and global metabolomics. I will come back to that. And we're also very interested in the bacterial composition of the gut. And this lab is performing all these activities. And finally, it's a large database structure with people working on keeping track on the data and doing the linkage to national registers to capture who got different diseases. And from these populations, we then we identify risk factors, and we identify back individuals based on those risk factors and try to do targeted interventions. And I will come back to what I mean with that. But as you have seen, we are very fond of genetics, as obviously that is the earliest sign or the earliest measurable risk factor if you have a mutation or a disadvantageous mutation for future risk of disease. This here is from my own thesis work in the 90s, where reason why I show this is because this is, some of you may know Little syndrome. It's a very rare form, which is a 100% genetic form of hypertension, where you get too many sodium channels in the distal tubules and collecting duct in the kidney, so you hyperabsorb sodium. Why I think it's fascinating, because it's a 100% genetic disease, but you can treat it with a 100% environmental action, which is sodium restriction, because if you restrict sodium, you will get this normotensive if you do it early enough in life. Then you may say, well, that's a strange example because this is a very rare disease. We have also looked at the common things. So this is a study that we did together with two other groups in the United States, where we measure the polygenetic risk for coronary artery disease. So the polygenetic risk is based up on hundreds of different gene variants. So you can have a low or a high number of all these various risk variants. And what was consistent between these three large population-based studies was that, as you can see, in people with high genetic risk, the bars to the right, if you have a favorable lifestyle on top of your genetics, of your polygenetic risk, you can basically eliminate that genetic risk. So we don't need any gene therapy or something like that to get rid of the genetic risk. But we need lifestyle changes, because these people are more sensitive to lifestyle, to bad lifestyle, but they also respond better to lifestyle. So I like this simplified figure that a high genetic risk of coronary artery disease is, of course, bad. And it means what that risk stands for. It means you're more sensitive to a bad lifestyle. The good side of the coin is that if you have that high genetic risk, you have more effect of your lifestyle changes. So if you manage to improve your lifestyle, you can get rid of most of your polygenetic risk for coronary artery disease. Another interesting thing with genetics is its capability to identify novel drug targets. This is an old story that I think many of you know, the PCSK9 story, but it actually began with genetics. This is also a joint paper from back in 2008, where we found that if you have a loss of function mutation that is relatively rare in the gene coding for the PCSK9, a key protein in cholesterol pathway, then you have a protection. The loss of function gives you protection from myocardial infarction in this case. So this indicates, well, if a genetic loss of function is good for you, then you could create a drug and attack that PCSK9, which is exactly what happened by various drug companies. So this is obviously a method that could work for other drugs as well. So we have therefore sequenced in one of these population studies that I mentioned, we have done sequencing, DNA sequencing of all coding regions in the genome, so-called exome-wide or whole exome sequencing in 30,000 individuals, because we want to find other things like the PCSK9 mutation that might help us to treat diabetes, to treat obesity, and so on. And we did actually identify, there are several findings from these collaborations, but one very interesting one, I think, is that if you have a genetic loss of function of something called G-coupled protein receptor 75 that is expressed in the brain, then you are markedly protected from getting obesity. And in animal experience, it's shown that this mutation, this GPR75, if you have loss of function, so you have less appetite drive, so you eat less, and you also have a higher basal metabolic rate. So from both these, you will get less obesity. And there are now companies developing GPR75 receptor antagonists. This is, of course, a long story to end up to our patients or even more in the population, but this shows the potential of knowing loss of function. We have another example where genetic helped us to find a new important endocrine system of involvement in diabetes, obesity, and cardiovascular disease. This actually came from that genetic variants in something called sortilin-1. It's a sorting protein that is expressed in many different tissues, including the liver and vasculature. And this sorting receptor is actually the receptor for a hormone, neurotensin. And neurotensin is a very important gut hormone that, so we know that a mutation or a common variant in the receptor for this hormone was strongly linked to coronary artery disease. So, and it does so both through affecting your levels of LDL cholesterol. It has to do with the lipoprotein sorting function of the neurotensin, and also GLUT4 increases risk for diabetes by having a defect in how the glucose transporter is expressed in the skeletal muscle. So in collaboration with Mark Evers in Kentucky, we found that mice that are knocked out, they don't have neurotensin. On the right-hand side, if you give these mice, normal mice, a high-fat diet or a Western diet, if you lack neurotensin, you don't develop obesity, or you develop much less body fat excess than in the wild-type mouse. And it turns out that the key role in obesity development of neurotensin is one, that you actually become more efficient in absorbing your fat from the intestinal lumen into the bloodstream. Very simple mechanism, but that's not all. It also puts the fat exactly where you don't want it. So neurotensin does two things, and this has been very elegantly shown both from the knockout model of neurotensin, but by other groups who have studied the receptor, the sortilin-1, that the whole story started with, that if you knock out the receptor or you don't have the hormone, you're protected from getting fat in your liver. But if you have a normal amount of neurotensin, your fat will accumulate in the liver and you will develop NAFLD. And this is clearly seen also in humans, that high level of neurotensin in your circulation is very powerfully linked to the likelihood of having NAFLD, or liver steatosis, non-alcoholic fatty liver disease. This is the latest paper that is not yet published on neurotensin, but of course, the whole story began with an association with atherosclerotic disease in humans. And these mice, if you in two different models of atherosclerosis, either gene therapy, so to say, with PCSK9, and on the right-hand side, the knockout of the LDL receptor. On top of that pro-atherosclerotic background, if you have mice who have or do not have the neurotensin gene, those who lack neurotensin have much less atherosclerosis than those who have normal level of neurotensin. So, like depositing fat in the liver, you seem to deposit fat in the vascular wall, leading to atherosclerosis. And it's also clearly shown in many different publications that the high level of proneurotensin or neurotensin in the circulation is a strong predictor in humans of future risk of myocardial infarction, stroke, and cardiovascular death. And in fact, this is one of our latest papers that if you have high levels of neurotensin, which is the risk is really somewhere around, if you're above 150 picomoles per liter in your fasted plasma concentration of neurotensin, you have a markedly increased risk for all-cause mortality. Well, and this is not surprisingly driven by cardiovascular mortality, and because of the liver, the NAFLD effect, also by hepatic pathologies. So it also gives you a shorter life to have high levels of this hormone. And why were we created with a hormone that kills us from the first place? It's because having a neurotensin, as I showed, is genetically very strongly determined. And tens of thousands or hundreds of thousands of years ago, and from a long time back, this was a survival advantage. It was good to absorb all the fat because you ate relatively seldom, and also to store your calories centrally in the abdomen and in the liver. At that time, it was a survival advantage. But as you all know, today we have a big problem with obesity and too much food intake and too little physical exercise. So today, this, we believe, is the reason why in today's environment, this genetic thing that was a survival advantage now kills us instead. Now, what can we do about this thing? Because we also say it's never any use to identify risk that we cannot treat, especially not in the population level. So one thing was there was a drug already developed that blocks the other main neurotensin receptor, the neurotensin receptor 1, that I haven't talked so much about. But since that drug was available, we tested it in mice, and it has a certain effect on preventing obesity, but it's not a massive effect. Plus, it goes into the brain and has a lot of side effects. So a more promising thing is to use monoclonal antibodies that block the hormone, that prevents the hormone from binding to both these two receptors. And that clearly works. We've shown together with a French group that these and neurotensin blockade with antibodies lead to enhanced weight reduction in obesity, and also gives you less fat in both in the central and peripheral compartments. So the problem is, of course, these monoclonal antibodies we have only used in mice. There is a human form under testing in certain conditions, but this will take many years because if that is coming out. So what we're trying now is actually to use orlistat. So orlistat is a lipase inhibitor, so that if you give orlistat, the sensor from the enterocyte that kicks out neurotensin is blunted, because if you have you have the lipase inhibition, so you don't get the fat into the enterocyte, and then you will have you can lower neurotensin concentration dramatically. And this, we believe, will both decrease, of course, the amount of fat uptake that is available for storage in the liver, but also to prevent this trafficking of fat to the central compartments, the central storage of this hormone. The final example is on how we can use metabolite measurements in blood to identify people who either eat well, they follow the guidelines on a healthy diet that is very similar to a Mediterranean diet, we would say, probably similar to some specific diets also in Japan, but it's much based on, it's not a vegetarian diet, but it's not so much meat, but there is meat, there is a lot of fruit and vegetables, beans, legumes, and fruit and berries in this diet. It's not a strange diet, it's a very normal, but we still have a problem of having people following it. So what we did, in these large cohorts, we had very detailed registration of what people ate during 7 days menu books. And we identified a profile, if you wish, of metabolites in blood, in fasted blood, that could very accurately tell us if you are adhere to the healthy diet, or if you do not, if you eat more like the lower picture down with a more, let's say, Western diet. So, but what was even more interesting, that profile of molecules that is much breakdown products of different food items, that profile also was associated in longitudinally during long-term follow-up with a markedly lower risk of getting diabetes or getting coronary artery disease. So we can measure by these metabolites both your risk of diabetes and coronary artery disease in the future, and it also tells us how you eat. And we did a small fun experiment where we had 60 Swedish people staying 1 week in a typical Mediterranean diet area. And each line is one individual's change. So the average change of this profile was a doubling of the level of the healthy food blood fingerprint. And 100% of the subjects who ate Mediterranean diet also increased. And like I said, this is a mixture of substances that come from food, that healthy food, they go up, and unhealthy food, they go down. And these levels are also associated with protection, or on the bad ones with risk for coronary artery disease. So we are now intervening. We're trying to make people, just like if you initiate antihypertensive therapy, of course, you will measure the blood pressure, and you would encourage the patients to measure the blood pressure to see the effect. It's the same thing here. If we initiate a diet intervention on this healthy diet, we measure how it goes in the blood. We believe that the compliance could be much better, because, as I think many of you are very well aware, it's very difficult to make people change their food habits. So I'm coming towards the end of this talk. This little guy is someone my wife and I met on a vacation trip recently in Thailand, and he seems to be eating very well. Papaya, I think it is. But what I wanted to summarize with is that we are pretty sure today that your genetic risk that some of our colleagues and many patients believe is non-modifiable. It is. A high genetic risk can be diminished by lifestyle. So we don't need gene therapy in high genetic risk individuals. You can use lifestyle, but you need have the people to perform the lifestyle change, of course. I also exemplified with loss of function mutations, with the classical examples being the mutation, the PCSK9 gene that led to the development of PCSK9 drug inhibitors. And there is another example that is an anti-obesity mutation, you can say, that we're hoping will result in a new anti-obesity drug in the future. But there are many other examples in the research world today with these loss of function mutations that protect from disease. So this is probably going to be an interesting area in terms of finding new drug targets. I also told you about the discovery of the neurotensin system that is genetically enriched to store fat. But in today's society, we have to do something about that because we eat too much. And we have monoclonal antibodies that might come in 5-10 years. Today, we're trying to, in certain individuals who have this genetic predisposition to use orlistat. And finally, a measure of healthy diet in blood with a blood sample, because it's commonly not enough to talk with the patients and say how they should do and get the feedback. We believe this objective fingerprint in blood might help the physicians and dietitians and nurses to guide their patients right. And as I began to say, we are really impressed by the Moonshot program and Moonshot grant, and together in collaboration between Lund University and University of Tokyo, we have started in Malmö, in one of these cohorts that I presented, a protocol that is very similar, almost identical to profiling that is being done here in Tokyo and Japan within the Moonshot program. So it's a great collaboration. I'm very happy that we have it. I know it's going to be generate successful new data, and we are also collective in a collaboration between Sweden and Japan, hosting a meeting in Okinawa starting in the end of this week. You can see the poster to the left, which is about healthy aging and many essential parts of what we have been discussing here today. These are people in my group, and you can see Dr. Svensson and Kishi Svensson here. This is just before the pandemic came. And I want to also acknowledge the funder that made our own research possible. Thank you very much for your attention.
Thank you very much, Professor Melander, for that excellent presentation and very impressive research that your group has performed. Thank you again very much, Professor Melander, for coming to Japan and for giving us this wonderful talk. And thank you to all those who attended, especially Professor Nangaku, who's here, and of course also Professor Tay. And thank you to everyone who asked very important questions. Thank you.
それでは本日のセミナーを終わらせていただきます。どうもありがとうございました。失礼いたします。